Welcome to For Science Salah Lotfy channel, where science meets curiosity. في هذه القناة لا نكتفي بعرض الأبحاث العلمية، بل نترجمها إلى المعرفة التي تصل إليك أينما كنت. Our mission is to make global scientific research understandable, accessible, and multilingual. جميع الفيديوهات العلمية هنا مترجمة إلى اللغة العربية، بالإضافة إلى لغات عالمية أخرى. French, Spanish, German, Urdu, and more. Because science speaks every language. نعم، العلم يتحدث بكل اللغات، ونحن نحرص أن يصلك بلغتك. If you're passionate about science, you're in the right place. اشترك الآن وابدأ رحلتك مع العلم خطوة بخطوة. For Science Salah Lotfy channel, العلم للجميع. Welcome to For Science Salah Lotfy channel, your go-to source for unraveling the complexities of the scientific world. We're dedicated to bringing you the latest breakthroughs, cutting-edge research, and in-depth explanations, all presented with clarity and precision. Join us as we explore the fascinating frontiers of discovery, where every question leads to a new understanding. Today, we're diving into a critical area of cancer research, exploring how scientists are targeting the very source of energy that fuels these deadly cells. In the relentless battle against cancer, one of the most promising front lines lies deep inside the cell, within the intricate machinery that powers its growth and survival. We're talking about the mitochondrion, often referred to as the powerhouse of the cell. These tiny organelles are responsible for generating the energy that cells need to function, and cancer cells are no exception. In fact, many cancer cells are heavily reliant on mitochondrial activity to sustain their rapid proliferation and resistance to treatment. A groundbreaking new study, spearheaded by the brilliant Dr. Mahmoud El-Shemerly, offers a beacon of hope in the fight against cancer. This research brings us significantly closer to developing effective strategies for defeating tumors by specifically disrupting their energy production pathways. It's a well-known fact that many cancer cells primarily rely on glycolysis, a process that breaks down glucose for energy. However, a significant subset of cancer cells exhibit a different metabolic profile, depending heavily on oxidative phosphorylation within the mitochondria to survive and thrive. This study sheds light on how to target this vulnerability. The journey to discovering new cancer treatments often begins with a deep dive into the fundamental science of how cancer cells function. In this case, the research team embarked on an ambitious project, screening over 110,000 compounds. Their approach was not just broad, but also incredibly clever. They employed a unique strategy, forcing cancer cells to rely heavily on mitochondrial energy production. This was achieved by growing the cells in a specialized culture medium where galactose was the primary sugar source, instead of the more common glucose. This seemingly simple switch had profound effects, making the cancer cells uniquely vulnerable and revealing potential therapeutic targets. Through this meticulous screening process, a promising lead compound emerged, initially identified as B001. Recognizing its potential, the team didn't stop there. They embarked on a journey of molecular optimization, carefully tweaking and refining the structure of B001 to enhance its efficacy and minimize potential side effects. This iterative process of chemical modification and biological testing ultimately led to the development of two significantly more potent drug candidates, B007 and B008. These optimized compounds showed enhanced activity against the targeted cancer cells, paving the way for further investigation. To assess the real-world potential of B007 and B008, rigorous preclinical studies were conducted using mouse models of breast cancer. These models mimic the complexities of the disease in a living organism, providing valuable insights into how the drugs might behave in human patients. The results were highly encouraging. The studies demonstrated that oral administration of these compounds led to a significant reduction in tumor size within the treated mice. This positive outcome underscored the potential of these compounds as a novel therapeutic approach for breast cancer. Further investigation revealed that the effectiveness of these inhibitors was particularly pronounced in tumors characterized by low levels of a protein called MCT4. MCT4 plays a crucial role in helping cancer cells remove lactate, a byproduct of their altered metabolism. Without sufficient MCT4, cancer cells struggle to manage lactate buildup, making them especially vulnerable to the effects of B007 and B008. This discovery highlights a potential biomarker for patient selection, suggesting that individuals with tumors exhibiting low MCT4 expression may be particularly responsive to these novel inhibitors. This research offers new hope, a therapeutic option for resistant cancers, showcasing intelligent drug design with orally available drugs that could reduce treatment costs. While balancing efficacy and safety remains crucial, this paves the way for personalized mitochondrial-targeted therapies and future combination treatments. As we conclude this exploration, remember that the fight against cancer is far from over. The journey continues, propelled by relentless research and unwavering hope. We are moving steadily toward personalized therapies, treatments precisely tailored to an individual's unique cancer profile. These therapies aim to target cancer from multiple angles, attacking the disease on various fronts simultaneously. The ultimate goal is to achieve more effective outcomes with fewer side effects, improving the quality of life for patients undergoing treatment. This is the future of cancer care, a future we are actively building. Thank you for watching this episode. We hope you found it informative and insightful. Stay with us at For Science Salah Lotfy channel, your go-to source for accessible scientific knowledge, where science is made accessible for everyone. We believe that discovery should never be lost in translation, and that everyone deserves to understand the world around them through the lens of science. Subscribe to stay updated on our latest videos and join our community of science enthusiasts.
Welcome to For Science Salah Lotfy channel, where science is made accessible and inspiring. Today's episode takes us into cutting-edge cancer research straight from Switzerland. The research we're exploring today is titled Discovery and Optimization of Oxidative Phosphorylation. Inhibitors from a phenotypic screen. This important study was published in the journal Anti-Cancer Drugs. It was received on April 25, 2025, and accepted after revision on May 23, 2025. It was conducted by a highly skilled team at Basilea Pharmaceutica International Ltd, located in Allschwil, Switzerland. Let's acknowledge the contributors behind this work: Dr. Mahmoud El Shemerli, lab head, cancer biology; Florian Richelet, Master of Science, who also served as the corresponding author. These two authors contributed equally to the writing. Other co-authors include Dimitri Robet, Dr. Claude Neuhofer, Alexandra Kunz, Luc Burry, Claire Heisler, Norbert Herrmann, Dr. Jochen Spickerman, Dr. Sven Weiler, Dr. Paul McSheehy, Dr. Lorenz Kellenberger, and Dr. Heidi Lane. All researchers are affiliated with Basilea Pharmaceutica International Ltd, a biotech company focused on innovative therapies for cancer and infectious diseases. Please note all authors were employees of Basilea during the study and may hold stock or stock options. The research explores a breakthrough in targeting mitochondrial metabolism, a powerful vulnerability in cancer cells. Using a phenotypic high-throughput screen, researchers tested over 110,000 compounds to discover new inhibitors of oxidative phosphorylation, or OXPHOS, a process cancer cells use to generate energy. The goal? To disrupt ATP production and oxygen consumption inside cancer cells, weakening their survival, growth, and resistance. One promising compound, called B001, was optimized into B007 and B008, both orally available and highly potent. These molecules specifically inhibit complex 1 of the mitochondrial electron transport chain, choking the energy supply of cancer cells. Animal studies showed significant reduction in tumor growth, particularly in breast cancer models with low MCT4 expression, a biomarker that may predict treatment response. However, some side effects like body weight loss were observed, highlighting the need for further refinement before moving to clinical trials. This study demonstrates enormous therapeutic potential of mitochondrial-targeted cancer drugs. By understanding tumor metabolism, scientists are designing smarter treatments, more precise, more potent, and potentially more effective. With MCT4 as a possible biomarker, we are moving closer to personalized cancer therapy treatments matched to each tumor's unique biology. For more scientific discoveries from around the globe, stay tuned to For Science Salah Lotfy channel, where research meets clarity. Like, comment, and subscribe to help us make science accessible to everyone, because science belongs to us all. For Science Salah Lotfy channel, where cutting-edge research meets accessible science. Today we explore an exciting frontier in cancer treatment, targeting the energy factories of cancer cells, the mitochondria. Cancer cells are notorious for changing how they generate energy. Traditionally, many tumors rely on a process called aerobic glycolysis, even when oxygen is available. This is known as the Warburg effect. But growing evidence tells us there's more to the story. Some cancer cells still depend on mitochondrial oxidative phosphorylation, or OXPHOS, for survival, growth, and even resistance to therapy. And this is where a new therapeutic opportunity opens up. Mitochondria are more than just the powerhouse of the cell. They play vital roles in signaling, cell cycle regulation, apoptosis, and metabolism. In cancer cells, mitochondrial metabolism allows them to thrive under stress, use alternative fuels like glutamine and fatty acids, and, importantly, evade drugs. This metabolic reprogramming is not just a side effect of cancer, it's a weakness waiting to be exploited. To find a way to shut down this mitochondrial survival system, researchers conducted a high-throughput, phenotypic screening of over 100,000 compounds. But here's the clever twist. They grew cancer cells in galactose instead of glucose. Why? Because galactose forces cells to rely on mitochondria for energy. So any drug that killed these cells in galactose, but not in glucose, was likely targeting OXPHOS directly. From this they discovered a new class of mitochondrial complex 1 inhibitors, molecules that block a key component of the electron transport chain. Through focused medicinal chemistry, two promising candidates emerged, B007 and B008. These compounds potently disrupted ATP production and reduced oxygen consumption in cancer cells. But that's not all. The researchers identified a key biomarker for drug sensitivity, a protein called MCT4. Tumor cells with low MCT4 expression were far more sensitive to treatment. That means doctors may be able to predict who will benefit from these new drugs. In mouse models of breast cancer, daily oral doses of B007 and B008 significantly inhibited tumor growth in cancers with low MCT4 expression. However, this success came with a challenge: body weight loss, indicating potential toxicity. This approach builds upon earlier efforts. Other compounds are already in patient studies. What makes this new research unique is its selectivity, oral bioavailability, and the use of MCT4 as a predictive biomarker. It offers a new pathway to personalized cancer therapy, one that targets the metabolic wiring of tumors themselves. This breakthrough reminds us, cancer is not just a genetic disease, it's also a metabolic one. And now, with precise tools, we can begin to cut off its energy supply. Stay curious, stay informed, and subscribe to For Science Salah Lotfy channel, where science comes to life. Welcome back to For Science Salah Lotfy channel, where scientific discovery becomes accessible and inspiring. In today's episode, we dive into the experimental engine room of a groundbreaking cancer study, where theory becomes practice and molecules meet biology. To explore how cancer cells respond to mitochondrial inhibitors, scientists began with HeLa cells, a cervical cancer line staple in biomedical research. But instead of growing them in glucose, the team cultivated these cells in galactose-based medium, forcing them to depend entirely on mitochondrial energy. Why? Because only then can you detect if a drug targets the mitochondria. Over several days, they gradually shifted nutrient balance from 50% galactose to 100% training cells to rely on oxidative phosphorylation. The next phase screened thousands of chemical compounds. HeLa cell lines and breast cancer cell lines were seeded into 96 well plates and 384 well plates, exposed to test compounds, and incubated for 72 hours. Cells were then stained with crystal violet, a dye that reveals cell survival. Absorbance was measured at 590 nanometers, showing precise cell growth inhibition. Some plates used YO-Pro-1 iodide to quantify DNA content under lytic conditions, confirming anti-proliferative impact. But how do we know compounds target mitochondria? The answer lies in two key tests, the ATP assay and oxygen consumption assay. In the ATP test, luciferase and luciferin were used, the same glowing reaction found in fireflies. If ATP production drops after treatment, it signals OXPHOS inhibition. Oxygen consumption was tracked in parallel using MitoExpress dye and specialized plate readers. Results clearly showed which compounds impaired mitochondrial respiration. To validate the platform, researchers compared results to known compounds. Rotenone inhibits complex 1. Antimycin A inhibits complex 3. FCCP uncouples the respiratory chain. Taxol and doxorubicin are traditional chemotherapy agents. Only mitochondrial inhibitors cause selective inhibition in galactose, proving the test worked as intended. The team used GraphPad Prism software to calculate IC50 values, identifying concentrations needed for 50% growth inhibition. Researchers then turned to immunoblotting to explore MCT4 protein levels, a lactate transporter in each cancer cell line. Why MCT4? Cells with low expression are more vulnerable to mitochondrial inhibition. Then came in vivo studies. Breast cancer tumors were grown in mice using MCF7 cell lines and BT474 cell lines. Tumor sizes were tracked, animal health monitored, and oral drug dosing effects recorded. Compounds B007 and B008 showed tumor suppression, although weight loss was concerning, highlighting need for dose refinement. Before clinical development, researchers evaluated drug behavior in the body. They tested liver microsomal stability, plasma protein binding, and oral bioavailability in mice. Using mass spectrometry and dialysis systems, they confirmed compounds were orally active, reasonably stable, with acceptable plasma-free drug levels. From cell cultures to live tumor models, this study presents a robust experimental roadmap for identifying mitochondrial inhibitors with therapeutic promise. It's a masterclass in targeting cancer metabolism and a hopeful step toward personalized treatments. Subscribe to For Science Salah Lotfy channel. Stay with us as we explore the science shaping tomorrow's medicine.
Welcome to For Science Salah Lotfy channel, where we explore the frontiers of research shaping the future of medicine. Today, we venture deep into a revolutionary cancer study that targets the very core of cellular energy, the mitochondria. In the heart of cancer cells, energy production is hijacked. While most tumors rely on glycolysis, even in the presence of oxygen, emerging evidence shows that many also depend on mitochondrial oxidative phosphorylation, or OXPHOS. Disrupting this pathway could cripple cancer cells' ability to survive, grow, and resist drugs. So the researchers asked, what if we could design compounds that selectively inhibit OXPHOS? Using a nutrient-sensitized screening strategy, scientists at Basilea Pharmaceutica screened over 110,000 compounds. They focused on those that showed potent inhibition of cancer cells in galactose media, forcing reliance on mitochondria, versus glucose media. This intelligent setup filtered out hits that affected mitochondrial function specifically. Known inhibitors like rotenone served as positive controls, while classic chemotherapies like Taxol were used as negatives. The initial hit, a thiadiazole urea compound called B001. B001 showed strong selectivity in inhibiting ATP production and oxygen consumption in cancer cells. Using mitotox assays, it was confirmed B001 inhibits complex 1 of the mitochondrial electron transport chain, the very entry point of the energy-generating process. This mode of action mirrored that of rotenone, a known complex 1 inhibitor, but with a more promising drug-like profile. But B001 wasn't perfect. It had issues: low solubility, high lipophilicity, and off-target interactions. So began the medicinal chemistry campaign. By replacing functional groups and reducing molecular flexibility, scientists developed B006, B007, and B008. B007 and B008 showed higher potency, lower lipophilicity, better aqueous solubility, and reduced off-target binding. These improvements boosted both lipophilic ligand efficiency and oral bioavailability. Next, researchers looked at MCT4, a lactate transporter. Why? Because OXPHOS inhibition increases glycolysis, and glycolysis increases lactate. Cells lacking MCT4 can't expel lactate efficiently, making them more vulnerable to OXPHOS inhibitors. Using data from the Broad Institute and protein assays, scientists found low MCT4 expression in breast cancer cell lines like BT474 and MCF7, and high MCT4 expression in MDA MB231 and HCC1937. This difference predicted sensitivity to B007. To validate the theory, mice bearing subcutaneous tumors from BT474 and MCF7 cells were treated with B007 and B008. The results: significant tumor growth suppression, dose-dependent efficacy, and tolerable side effects when doses were adjusted. In MCF7 models, B007 and B008 reduced tumor size by up to 74%, but initial weight loss required dose adjustment. In BT474 models, both drugs worked well with fewer side effects. What about pharmacokinetics? B007 and B008 showed good absorption, high oral bioavailability, 54 to 72%, free drug fractions of 3 to 4%, and medium to low liver clearance in both mouse and human microsomes. This means they're not just effective, they're druggable. Like all powerful therapies, these compounds have risks. In some cases, weight loss was linked to treatment potency. Other mitochondrial inhibitors like IXS010759 have shown dose-limiting toxicities, including neurotoxicity and elevated blood lactate. That's why optimizing the therapeutic index, the balance between efficacy and safety, is critical. Combination strategies, especially pairing OXPHOS inhibitors with MCT4 inhibitors, could allow lower doses and better safety. This research lays the foundation for a new class of cancer treatments, mitochondrial complex 1 inhibitors. With the identification of biomarkers like MCT4, doctors could one day tailor treatments to those most likely to respond. Still, more work lies ahead: long-term toxicity studies, combination therapies, and clinical trials in humans. But the message is clear: targeting cancer metabolism is not just theory, it's becoming practice. Thank you for watching this deep dive on For Science Salah Lotfy channel. If you found this content insightful, like, share, and subscribe for more science made simple. Together we bring tomorrow's medicine to today's understanding.
Welcome back to For Science Salah Lotfy channel, where we dive into the science shaping tomorrow's medicine. Today's video continues our journey into a remarkable study on cancer metabolism, focusing on the progression from discovery to preclinical validation of mitochondrial complex 1 inhibitors. This research, titled Discovery and Optimization of Oxidative Phosphorylation Inhibitors from a Phenotypic Screen, was published in the prestigious journal Anti-Cancer Drugs. It was submitted on April 25, 2025, and accepted on May 23, 2025. The study was conducted entirely by a dedicated team at Basilea Pharmaceutica International Ltd, based in Allschwil, Switzerland. Let's recognize the outstanding authors behind this work. Dr. Mahmoud El-Shemerly, Lab Head, Cancer Biology; Florian Richelet, Master of Science and corresponding author. Both authors contributed equally to this work. Other co-authors include Dimitri Robet, Dr. Claude Nuoffer, Alexandra Kunz, Luc Brewery, Claire Heisler, Norbert Herrmann, Dr. Jochen Spickermann, Dr. Sven Weiler, Dr. Paul McSheehy, Dr. Lorenz Kellenberger, and Dr. Heidi Lane, all affiliated with Basilea Pharmaceutica. All authors are or were full-time employees of the company and may hold shares or stock options. The researchers used a robust experimental framework. They grew HeLa cell lines in galactose and glucose conditions. They performed crystal violet assays for cell proliferation, quantified ATP using luciferase-based luminescence, and monitored mitochondrial oxygen consumption with MitoXpress. Through these methods, they identified selective inhibitors that disrupted mitochondrial respiration, namely B001, which was later optimized into B007 and B008. These compounds were validated with immunoblotting for MCT4 expression in breast cancer cells, comparative sensitivity analyses across multiple cancer lines, and reference compounds like rotenone, FCCP, Taxol, and doxorubicin to benchmark effects. The compounds underwent rigorous ADME profiling. This included microsomal stability in mouse and human liver microsomes, plasma protein binding via rapid equilibrium dialysis, and bioavailability measured after both oral and intravenous administration. B007 and B008 showed oral bioavailability exceeding 50% and favorable drug-like properties. The final test involved xenograft tumor models using BT474 and MCF7 breast cancer cell lines in mice. B007 and B008 demonstrated substantial tumor volume reduction, dose-dependent efficacy, and acceptable side effect profiles when dosing was optimized. The presence or absence of MCT4 expression played a key role in treatment response. This research highlights the promise of OXPHOS inhibitors in personalized cancer treatment. While challenges like toxicity and selectivity remain, these results are a major step forward in the fight against drug-resistant tumors. Stay tuned for more on how metabolism-based therapies are redefining the landscape of oncology. Thank you for watching For Science Salah Lotfy channel. Subscribe, like, and share to stay informed about breakthroughs that could shape the future of medicine. Because when science is clear, knowledge becomes power. Welcome to For Science Salah Lotfy channel, where knowledge knows no boundaries and research finds its voice. Today, I would like to pause and extend my deepest appreciation, not as a presenter, but as a scientist, a colleague, and a grateful admirer of outstanding scientific work. It is my great honor to extend sincere thanks and recognition to the brilliant team behind the groundbreaking research titled Discovery and Optimization of Oxidative Phosphorylation Inhibitors from a Phenotypic Screen. This study, recently published in Anti-Cancer Drugs, stands as a shining example of how dedicated scientific minds can push the frontiers of cancer therapy by targeting mitochondrial metabolism with unprecedented precision. To the lead authors, Dr. Mahmoud El Shemerly, Lab Head, Cancer Biology, and Mr. Florian Richelet, Master of Science, and to every co-author and contributor, Dimitri Robet, Dr. Claude Neuhofer, Alexandra Kunz, Luc Bury, Claire Heisler, Norbert Herman, Dr. Jochen Spickerman, Dr. Sven Weiler, Dr. Paul McSheehy, Dr. Lawrence Kellenberger, and Dr. Heidi Lane. I thank you all on behalf of myself, Professor Dr. Salah Lotfy of the Egyptian Atomic Energy Authority, and on behalf of every follower of the For Science Salah Lotfy channel. A special word of appreciation also goes to Basilea Pharmaceutica International Ltd, based in Allschwil, Switzerland. Your dedication to scientific innovation, your investment in rigorous drug discovery, and your unwavering commitment to improving global health do not go unnoticed. You exemplify the best of industry-led biomedical research, and we are proud to present your work to a wider audience. To our valued viewers, scientists, students, and enthusiasts watching this video, your curiosity fuels this channel, your comments help us grow, and your passion inspires future episodes. I personally invite each of you to share your thoughts and insights on this important research in the comment section below. Let us start a conversation on cancer metabolism, on OXPHOS inhibition, and on the future of targeted therapies. At For Science Salah Lotfy channel, our mission is clear: to bring you the very best of global scientific progress, explained with clarity, translated with care, and delivered with purpose. Thank you once again to the brilliant team at Basilea, and to every follower of this growing community. Until next time, stay curious, stay engaged, and stay tuned for more breakthroughs from the world of science. For Science Salah Lotfy channel, science that speaks to all.